So, good morning, good afternoon, good evening, depends on where you are in the world now. So, thank you very much for joining us today for another exciting CASS talk. So, today we have the pleasure here to have Professor Jan Madsen from Technical University of Denmark. But today we are here to listen to the talk that will be done by Professor Jan Madsen about biocomputing, computing with biological circuits. And Jan Madsen is a full professor in computer-based systems and leads the embedded system engineering and its deputy director in the Department of Applied Mathematics and Computer Science. He holds a master in electrical engineering in the same university, and also as well a PhD in computer science in 92, also in the University of Denmark. And his research interests are in the intersection between computer science and biotechnology, with a special focus on design, modeling, and construction of microelectronic, MPSOC, and IoT, microfluid lab on a chip, and microbiological molecular computing systems, including development of design automation tools and design methodology. In these fields, he has co-authored more than 200 peer-reviewed journal and conference papers, 3 books, 13 books chapters, and 3 patents. He has co-supervised 47 PhD theses, and he was the general chair of DATE 2018, NOX 2012, Codes 2001, and the technical program chair of DATE 2007, Codes plus ISS 2011, Codes 2000. Dr. Madsen is a board member of EDAA, member of the Academy of Technical Science and its Council for Technology and Society. He was national ICT expert for European Horizon 2020, and he's a member of IEEE and ACM. So thank you very much, Jan, to accept our invitation. And the floor is with you to start your talk in a very exciting subject. So thank you.
Thank you very much, Ricardo. So thank you for the invitation and for the presentation. So what I want to do is tapping into computing from a completely different angle, but still trying to explore the things that we are used to. So just to be sure, I want to come with this disclaimer, because sometimes when I give the talks like this, people are misunderstanding. So biological circuits are not to replace silicon circuits or electrical circuits. It's circuits that has a different purpose, and this is what I want to try to elaborate a little bit on. Before starting on dipping into it, I just want to recap a little bit about computing as such. So we all know the Turing machine, where we have this infinite tape of symbols that we can manipulate through a head that can read and write on the tape, and some control into that can define functions. So that means in the tape, we can identify parts that describes or represent the type of function we want to work with. We can have places where we have some input, and then we may have other places where we put in the output from this function. Now, why I put this up here in the beginning will be clear later on, but this is just like the foundation of computing and the way we are building machines. So from the very first silicon computer, as you could say, this is one of the very first integrated circuits, building with just a few gates, 6 transistors, and even though it was quite simple at the time, this was the breakthrough that took us to the level where we are today. But even with very simple computing, putting it together, building out of, I think it was around 4,000 of these small chips, was enough to build a navigation system to bring the first man to the moon. So even for simple things, we can get very powerful result out of this. Now, looking into it, working at the very core level, this was kind of where I started back when I did my PhD in trying to figure out systematic ways of doing layout synthesis from a description, for a high-level description like the logic. And this is pretty much still what we are doing. So we can work with these logic gates, describing how zeros and ones are interacting in order to produce logic outputs of those. And the way that we are working with this at the logic level on circuits, like a simple inverter, is of course that we have a transistor diagram that defines how the current is running in this circuit in order to make the switching that we want to do. So this is just put out here as a simple example. So when the input is switching from low voltage to high voltage, then the output is in reverse in this sense. And the charging and discharging of internal nodes is actually delaying the shifting, and therefore you can see we see this change in ways, and then whenever the voltage is going through half the full voltage, it's switching from a 1 to a 0, and in this case again back to a 1. And we have learned to work with this abstraction in a very nice way, because we can have a region where we are sure that we can interpret this as high level, as a 1, and a region which is interpreted as a low level. And what we are working with in this part is mainly thinking about or measuring the concentration of electrons we have in the wire, or at a certain place, determining whether it's a 0 or a 1. And we also learned that we can put these gates together in doing cascaded logic, and we can actually infer the logic of the cascaded composition of those just by inferring the logic of the simple components into it. So I guess this we all know. This is basic stuff we learn in the university, at least if we are doing electrical design. And what I now want to is trying to take this a little bit further in a different direction. So when we look at circuits based on electrical based on the flow of electrons in a wire, this is how we get the flow of information. So actually having an electric field across a wire will actually push the electrons in one direction, and this is how we can move the zeros and ones. And we all know that this has been a very nice way of doing circuits for very, very long, and still is. But it can also get into the point where we can start asking the question that could we use other substrates to build logic circuits and start reasoning about things? And what I want to try to address is this question on how would this type of thinking of moving information, how would that work in biology? So what do I mean by this? So biological circuits can be defined as molecular circuits that are operating in the cells, consisting of molecular species. So that could be genes and proteins and small molecules and so on, that interact with one another in specific ways. And one of the examples, and that's the one I will show in this presentation, is that when we have a certain DNA sequence that represents a gene, or represents the code for producing, let's say, a specific protein from the gene, this gene is transcribed to produce a corresponding messenger RNA, which in turn then can be translated into the string of amino acids that makes up a specific protein. So inside the cells as such, we have these gene regulatory networks, and it's actually possible to tap into that to reprogram the way that they work. And this is what I want to try to unfold in this presentation. We can actually go one step up from this, where we're actually considering building cell circuits. So that means that we think of cells in a much higher level. So it's not what's going on inside one cell, but actually in a colony of cells. So we can look at cells being in different states and different types, or even from different species, that can signal one another and to control each of others' properties like growth, death, proliferation, and differentiation. And in this, the key variables in such circuits are the concentration and location of each type of cell. In this presentation, I will focus on the first part of it. So how to build circuits inside a single cell, because that's kind of like the basic start for the rest of it. And I will just say a little bit about the cell circuits. Now, if we look into to the flow of information that I was saying in our electric circuits, we have components that are able to do different functions, and to link those together with signals, we use wires, and the wires encapsulate the electrons and keeps them inside of this, and we can safely transport them from one end to another end. Now, in biology, the flow of information would tap into a completely different way of looking at the basic components in the system. Because here we are basing it on molecules, and molecules that are moved by Brownian motions. So that means it's kind of like the temperature that gets the different molecules to vibrate, and that moves them around. And once in a while, they bump into each other. And when they do that, something happens, or something may happen. And so these are very different. We cannot really encapsulate them into specific regions where there is a flow of information and other regions where there are no flow. So how do we take that into account when we want to try to build the circuits? So let me just say, what do I mean by building circuits in the biology? So this is an image of a E. coli bacteria. And what's going on inside this, all these, you can see this many different points and parts which are different proteins inside the cell. All of these are vibrating, bumping into each other, and things are happening. The yellow part in it is the DNA of the cell, and that is the one that we want to tap into. But if we do an abstraction out of from this biological circuit, and then just for a moment forget all the messy stuff going on inside the cell, we can actually view this cell in having three different, let's say, functions in it. As a function which is working as a sensor, and in that sensor we may get proteins from outside or proteins produced inside the cell in a certain concentration. So in this case here we have blue and green proteins, and when they appear in the right amount of concentration, then it may trigger the cell to do some responses on this. And this triggering of response can be described as logic gates in the circuit. So we can actually build based on the logic inside the triggering logic onto the concentration of different proteins produced inside the cell. And the outcome of this could be an act where it actually then produces another protein, like the red one, that is released into the environment. Just like electronic circuit, we need energy to do this, but energy is provided to the cells through some small energy molecules, and this is just the fact of life, how cells are working and balanced. So that's, in one sense, is an easy part, but it's also a difficult part in the sense that this is very well balanced. So it's the cell can do exactly what it's supposed to do, and if you want to have it to do more functionality, you may exhaust it, and it may actually die eventually. So even though some of the things that I will show you are in principle relatively simple, in practice to get it to work is very, very hard. Now, in what we can produce, it may not just be molecules or proteins that interact inside the cell or is released to the outside. We can also produce small molecules that the cells are normally using to communicate with each other, to do things like figuring out how many cells are there in the neighborhood or other things. It's actually possible to tap into this and control and modulate this communication, so we can program not only the cell itself, but we can also program the communication between the cells. Now, before we go into how this works and what it actually means, I just want to give you an idea of what type of systems we could build with this, and what people are looking into. So we could say that if we can change the code of a cell, what would we like to do with that? So we could take the very, very simple one, simple from a computational point of view, is what we will call the biofab things. So that is principle doing a program that forever will produce a certain protein. So that is a kind of a factory of producing, for instance, insulin or some other materials. Now, to give you an example of this, I have a colleague here at DTU, Professor Irina Borodina, who is working on cell fabrication and trying to modify specific cells to be good at producing things. And I just want to give you a kind of a interesting example of how you can do this. I mean, producing insulin is, of course, useful and an important thing. But in this project, what they are looking into is the insect pests that may harm crops, as in the image. One of the problems in getting rid of this is that we typically use chemicals to fight against them, but the insect pests are always one step ahead to overcome these chemicals, and we have to either produce more of it and getting it more toxic and so on. We may turn into these insects into being resistant to this, and then we have, a lot of toxic flowing around in the environment, which is not a good thing. So what Irina and some of her colleagues were considering is there a different way of doing this? And looking into it, instead of killing the insects, they wanted to prevent them from reproducing. If they cannot reproduce, they cannot spread, they cannot multiply. And the way they wanted to do it was producing female pheromones from these insects. So what does that mean? Well, it means that they tried or succeeded in actually re-engineering yeast to produce these female pheromones. And when they are in the environment, the insects get confused. They cannot find the females. And if they cannot find the females, they cannot reproduce. So it's a different way of fighting insect pests. By confusing the insects to not multiply, and it's now being turned into a product. So you can think of producing many different kind of things out of just getting cells to produce something. Here are some other examples that I took out of a presentation by Chris Voigt from MIT. He's a professor working on synthetic biology. We can produce different types of materials and chemicals. I showed you one chemical. Here's another one where it's a material by just having 4 genes in the E. coli always expressed. So it's just producing this amount. We can work with this into agriculture, making sure that the product stays healthy for longer. And it can also go into some parts of therapeutics, and I will come a little bit back to that. Now, we could take the problem one step up, and instead of just doing something, then reacting on something, and this would be then a biosensor. So in this case here, I show that if the yellow protein is present, then do a green fluorescent protein. Things like this can be used in healthcare for diagnostics and therapeutics. So on the left-hand side here, you see something where you take out a blood sample, and depending what is of molecules that are in this one, it triggers some reaction that changes the color. Or it could be some way of doing real-time health tracking by having bacteria reprogrammed in a pill, understanding what is going on, or analyzing things in different ways, like in the bottom part where you have like feces, and you try to see the reaction with it depending on what are the composition of it. You can also do therapeutics into treating a specific cancer part, so identifying whether you have a certain cancer cells in your body. Many different ways of actually using these sensing devices to monitor what is going on. Now we can go one step further, and this is where I think it becomes even more interesting, and this is what I call biocomputing. So that is actually then now not just reacting on something, but as combining the two. So if certain conditions are there, then trying to produce a response to that. So it will be sort of like a function triggered by certain inputs into this. And to give you a feel of what that means, there is a very interesting application produced by the group of Professor Fossenegger from ETH Zurich. This part is a paper that is now some years old. The thing they looked at was looking at psoriasis, and whenever you have a lesion of psoriasis, you get these red itchy spots. Now, actually, when this happens, there are some biomarkers indicating that you have this inflammatory condition. And their idea was to re-engineer a specific cell, to actually being triggered by these biomarkers. And in the case, there are two biomarkers that when both of them are present in sufficient concentration, that would be an indication on an inflammatory condition. And the cell in the area measuring this would then react on it, producing two other proteins that works as anti-inflammatory drugs. And they showed that in a controlled lab experiment in a petri dish with tissue having psoriasis, then by adding these cells, they could actually mitigate the inflammation. And looking into it, it's actually quite simple program, right? It looks at least from the beginning as a very simple thing. So if the two conditions are present, both of them, then you produce the two other proteins to work as drug. And if nothing is, or if they are not present or not in a sufficient concentration, then you do nothing. There has been a number of such breakthrough throughout the time. Now, giving you the sort of like this introduction to what kind of types of circuits we could build, then now let's go in to see what then is the technology of these biological circuits. Now I want to go back to the Turing machine and the identification of it like this, because if you look a little bit into this and you know a little bit about biology, then this tape actually looks pretty much like the DNA in the cell. And in the cells there are protein molecules that are forming parts that actually work as these heads that can work its way through the tape and make actions based on what it identifies. This is also known as the central dogma of life. So, and I'll now explain how this machinery of life works. You may be familiar with it, but if you have like a long strand of DNA, then we have some specific molecules, large molecules called RNA polymerase, that can attach to certain places on this DNA. When they attach to it in the right place, they start moving forward. And what it's doing is that it's splitting the double helix and making a copy of one side of it. So while it's running across this one, it creates a long string of a copy of a single-stranded RNA. And at some point, it gets to a point where it's detached from the DNA and also detaching this green line here, which is the messenger RNA. That one floats around, and then another big molecule can attach to this one. This is the one called the ribosome, and it would start reading that one. It would read then 3 bases at a time, and for each of those, there is a corresponding amino acid. So while it's reading across, it's building together a long chain of amino acids that when they are released, they curl up and becomes the structure of this protein that was encoded in the DNA. This is how it works in principle, and by this one we have created one molecule. But just like we have in regular computation, you can have multiple of these going on at the same time, pretty much like a pipeline execution. So we can produce each of these parts where this RNA polymerase attaches and detaches. We can have multiple stages of transcribing it at the same time. So in that sense, we can create a larger amount of proteins at the same time. Let's then look into exactly what's going on in here. So in the top, I have the DNA, and these are consisting of the nucleotides, and there are some different regions in here. So there is a region which is defining the gene, so that is what is turned into the protein at the end. And then the gene is whenever the RNA polymerase is transcribing it, is turned into this messenger RNA, which is a copy of one side of the DNA, as you can see in the middle here. And when getting to that point with the messenger RNA, then there are this ribosome that attaches to it and read it and stitch together this chain of amino acids, which you see here, which then in the end curls up as the green protein that I've shown out here, which is a number of these amino acids put together. So these are the principles of it, and we can capture that in a more symbolic way as having the line at the bottom being the DNA. We have the green arrow kind of here is the gene sequence. We have a terminator in the end, so that's a part of the DNA that describes where the RNA polymerase has to release it. And then we have some binding sites in the beginning here with the promoter that tells where and when the RNA polymerase can attach to it. And then there is this half circle symbol, which basically is determining the binding site of the ribosome. So you can see there are some parts of the DNA and RNA that are used to guide those molecules to where they have to read, but the essence is to turning the green part of the DNA, which is the gene, into the sequence of amino acids. Now I will use this symbol that we have in the bottom, so that's called SBAL notation, so synthetic biology open language, and one of the things that have made all of this possible is that people have been studying snippets of DNA and trying to understand by sequencing, by analyzing them, what kind of functionality they are actually producing. and building up data sheets of this. So in this case, it's something where you can see that depending on whether you have, you would either produce a low input rate, that is the open circles, and the filled circles is where you actually have a high rate of molecules produced over time. Now, these can be done, as I also explained previously, to work with building logic components. So what we are tapping into to build these logic circuits is the gene expression. And if I look at this symbolic explanation where I have this first arrow is the promoter, then we have the gene sequence, and then we have the terminator. Then the polymerase attached to this, it reads until the end. And then while it's reading the gene sequence, it's producing this mRNA sequence at a certain rate. Now, as always in biology, whatever you produce doesn't rest forever. So there is a decay where this molecule would not work anymore. Now, when it is there, when the messenger RNA is there, the ribosome can make the translation, and it can actually do that multiple times. So from one messenger RNA, we can create several proteins out of it until it's decaying and we cannot do it anymore. So we have here some production constants with rates, and we have some decay of these molecules also with some rates. And that actually taps into being able to describe this with linear differential equations, and we can start simulating and working on these things. Now, what I want to do is to go one step further, because we can also control how this works. So looking into this basic components, there is this DNA binding site. So that is where the RNA polymerase is attaching. That can be controlled by some other molecules. What do I mean by this? So this red molecule can be a molecule that either repress the expression of gene. That means that it prevents the RNA polymerase to attach to it. So if there are green or red molecules in the area, that would prevent the RNA polymerase to attach, and therefore we will not get the green protein produced. These can also work as activation. So that means if they are present, then they actually enhance the process of producing this gene sequence. We can go one step further, because the activation of this red molecule can be dependent on some inducers. So that means that, and these red one we call transcription factors, because they are regulating the transcription. So the transcription being the part where the RNA polymerase is reading the DNA to produce the messenger RNA. So here in this case, whenever the little blue molecule is attached to the red one, it gets activated either as a repressor or an activator, depending on what molecule the red one is. But this blue one can come from outside with small molecules that can penetrate the cell membrane. So this is a way that you can actually trigger the logic in the cell from the outside, and that is possible. So let's look at, well, based on this foundation, what kind of things can we actually build? What kind of circuits can we build? So we can do sensor gates. That is basically something that is activating. So if the molecule A as a transcription factor is present, that will enhance the production of the green protein. That would work as sort of a buffer or an enhancer. And it means that over time, we will have that the more blue there are, the more green will be produced. And if the green one is fluorescent, then we can see a response. That was kind of the sensor that I explained earlier on. I can also build other things, because I could actually then have the blue molecule being a repressor. So that means that if there are blue molecules in the area, they will prevent the RNA polymerase to attach, and hence they will prevent the gene expression to take place. That turns into become a NOR gate, or a NOT gate, or an inverter. So if there are very little blue, you would have green, and as soon as blue is increasing, you would decrease the amount of green proteins. So now you can see that it's kind of similar that we have in circuits where we are looking in the concentration of electrons, but here it becomes a bit more noisy, because the amount of concentration you need for a certain protein to trigger the circuit will depend on what type of circuit you're building and what type of proteins that you are using. I can put things together. So here's a way that I could actually create a NOR gate. So now I have two different transcription factors, A and B, the blue and the red one. And if none of them are present, then I generate the green one. So that is what you can see at the right-hand side. When they are low, we have plenty of the green one. Whenever they start, one or two of them to repress, then the production of the green one will be reduced, and the amount of green molecules will decay over time, and you will not produce any new ones. So we have here a simple way of doing a NOR gate. What else can we do? We can actually build toggle switches or flip-flop gates. In this case, it's two circuits that are both repressors, and they are triggered by the inducers. So by having either inducer 1 or inducer 2, we can actually shift the state of this circuit, and it will keep its states in either one or the other one. And we can use that to mimic the way of we having zeros and ones stored in the system. And we can use that to build some simple form of memory elements. Now, we can take this a bit further, because I've shown you now some very, very simple Boolean logic components. I've shown you the toggle switch to do memory. We can actually combine these two to build finite state machines, not big ones, but small state machines. We can do the sensors, the biomarkers that trigger the logic, and actuators, as I've seen. Now, in the beginning, I mentioned that besides doing single cell function, we can actually do multi-cell colonies and having function across this. So this can be used to produce more large computations. It can be used to mitigate noise in the system, because they are not very, and I'll show you in a minute examples of this. They might not be very stable. It's a noisy environment, and we can use it, as already explained, as cell-to-cell communication. Now here's an example taken from a very simple state machine with two states and some transitions turned into a gene regulatory network representation that then can be implemented as a composition of these biological circuits. And then you can have each of those going into a cell, and then you can see what happens and how the cell colony forms, and the states of the different cells are triggered by the share of these small inducer molecules. So it's possible to model such systems from single cell systems to multicellular systems. Now, when I want to design this, I can take pretty much similar approaches as we are doing to when we are designing circuits. So in this case here, we have a design build test phase. So I come up with let's say the function, the logic function I want to implement. I go into a design of it. That means that I try to figure out what are the right logic components to be put into that. Then I build the circuit. I build the circuit by stitching together DNA sequences that are representing these logic functions. And then I am doing a simulation of what is going on in that, trying to validate whether or not it works as expected. If not, then I do another iteration, but at some point in time, it may turn out to be the right thing, and then I have the DNA sequence that represents this logic function. Now I can take this a bit further when building and trying to figure out. We can do technology mapping as we are used to. You can have a number of these different gates. So in the electric circuit, we can replicate the same type of gate. Here we have to use different gates because if you see it here, you have a number of inverters, each triggered by specific molecules. And then you have to find mappings between those so that the molecules produced from ones can actually be used as input signals to another one, as shown in the bottom here. Now I can use pretty much the same stuff that we do in trying to minimize and do the technology mapping. So if I have a function as the one in the top with C plus the negation of A plus the negation of B, I can look into the negation of B. I can have two different inverters and the output of those fitting into one of the ones in the red area of the NOR gates. So I can build something up, and there are different components I can map in depending on what I already have used and how well they adapt and match each other. And then I can build from that the circuit diagram that most likely will be the one to work. And then I can simulate it. And in the beginning, I mentioned that all this, the rates and decay and so on, is a matter of writing differential equation. And this is usually how we do it. But with ordinary differential equation, it sort of assumes that we have enough molecule that it's okay to average over a large amount of molecules. But when we zoom in and work inside the cell with these molecules, it becomes much more stochastic. And this is what this simulation is showing. It's the concentration over time. And you can see it's not nice bended curves, but it's actually some jiggery things going on. But looking into this and analyzing it, we can try to infer what are the logic signals of each of these signals in different places. So how many of a certain molecule, the red molecule, does it take to trigger the function from a 0 to a 1? And by doing this, we can play around with stuff. What we did in the beginning when we were working with this was to build a simulator, a dynamic simulator that's ran over time, and we could sort of try to change the concentration on some knobs. So we turned let's say the logic description of the network into a simulator, and then we could over time turn up and down on different levels and see how the circuit triggered based on that. That was a very convenient way of getting some feeling of what was going on. But when it becomes stochastic, it becomes much more difficult to actually see whether or not it would work in practice. And for that, we have looked into using statistical model checking to try to reason about probabilities of the circuit to actually within some parameter span actually producing the right functionality or not. In this case here, it's a NAND gate that is evaluated. So there is the green one is sort of like a multiple of many simulations, but with statistic model checking, you can sort of like infer a probability of it to work without having to explore the full space. And that's a very convenient way to understand like what are sort of like the limits or the span of the rates that can be that needs to be upheld in order for it to work in a correct way. Lastly, I want to just tap into what happens then when you have this circuit. You have the DNA. You can get the DNA as a sequence in your computer, mailed to a company that can then actually 3D print it as the DNA sequence. And you get that DNA sequence back. And what you want to do typically is then taking that DNA, doing a PCR where you do thermocycling and you upscale the amount of DNA you have. Then you have a protocol like the user cloning that stitch these DNA parts together. When you have them together, you want to do the transformation. So that means getting the new code into the cell and getting it booted up with that code. And then you look into the Petri dish to see whether or not the cells behaved as they should according to the programming you put into it. And you would see how many of them worked out correctly and so on. This is a fairly standard way of doing it in the lab. But we have been working on trying to also scale this one down. So instead of working with test tubes and pipetting and so on, we have explored the possibility of doing liquid computing. So by that means is like here. So the test tube is just a small drop, and the pipetting and moving things around can be done by electrodes, where you have an isolating surface and then some typically silicon oil that makes it very hydrophobic. So you get a perfect circle lying there. And by applying a field across it, you collapse sort of one end of the surface tension of the drop, and it rolls over to the next electrode. And by turning on and off things like this, you can get them to move around. This is the system that we have built. Is a small computer system. You have at the right-hand side, you have the chip. This is where we manipulate drops, and there is some control to turn on and off each of these electrodes that are inside the chip. We have here 640 electrodes that can be individually turned on and off. We can program temperature zones on it, so we can move drops in and out of temperature zones to do PCR. And we can do some volume measurement to understand exactly where the drops are and whether they move as they should. At the left-hand side is a small computer microcontroller control system that controls the actuation of the sequence of electrodes that can be loaded in as a program or can be in, let's say, wireless connection with a standard computer. And then in the middle is the power supplies, where we boost ordinary batteries to 200 volts, which is what we need to get the drops to move. Now, in this one, it's shown as an open space, but normally we would like to keep the droplets in a sandwich structure. So we have a glass plate on top of it, and inside that we can move the liquids captured inside a silicon oil. And then when we do it like this is how it looks. We can move around small amount of liquids. So these electrodes are 2 by 2 millimeters. So we have roughly a drop filling one is in the order of 20 microliters. And you can see that we can actually move them around. We can mix them. And now we're showing here that we can actually also split them again. So we have operations that is basically what you're doing by pipetting in a regular lab. Now this can be done in a very programmatic way. And if you look at it, there are some operations of the droplets. I've shown the move, the split, the merge. The mix is just like when they have been merged together, you move them around. And we can also pinch out small drops from reservoirs to keep on loading in things. And then we have some functional nodes where we can heat and cool the drops. We can load them in or extract them. We have sensors to understand what or measure what is inside the drop, so we can see if reactions happen the way that they should. And we have things like magnetic beads, magnetic field to purify, so we can have magnetic beads that are capturing certain molecules. We can grab them and drag them to the bottom. We can move the liquid, and then we have removed those molecules from within the drop. This allows us to actually do a full tool chain, and what we're working on is having a full software stack on top of this, where we can do PCR. And PCR is one of the stages in this cell cloning process. Now, what we are doing is having just like in a regular computer, we have these operations. We do an assembly language in which we can express things. We have a virtual machine, so we can capture in real life what is going on and make decisions about changing the protocols while the experiments are running. And what does that mean? And just to show you an example of it, here it shows an example where we have a camera, and the camera is detecting the color of the drop that goes beneath it. You can see it's identifying the RGB color code, and depending on the result of that, it decides on doing that the next step is one of several protocols that actually moves the drops according to its color to certain parts of the chip. So we have a possibility to control everything, monitor things while it's going on, making decision on how this would work. So it means that what I've showed to you about building these biological circuits, we're working with getting from the logic functional description into a design phase where we explore in the simulator what is the most likely case for a DNA that would actually turn the cell into this functionality. And usually it's not just one; it may be several that we can try to investigate. And when we have this, we also then now have this small lab that can actually stitch all of this together, assemble the circuits, and get them to work by cloning the cells and getting them the new DNA into the cells and getting them into the petri dish for understanding whether or not the circuit works and behave as planned. So I hope with this I give you a crash introduction to what biological circuits are and how you can use them and why we want to work with those. So just to wrap up, there is a book. We did a book on genetic design automation where some of this stuff is explained. And then for anyone being interested in the field and getting a very good introduction into it, I can recommend this book, The Genesis Machine by Amy Webb and Andrew Hessel. And this is a quote from Andrew Hessel saying that the promise of synthetic biology is a future built by the most powerful, sustainable manufacturing platform humanity has ever had. So a completely different way of looking at digitization and building circuits to compute than in the classical sense. And this brings me to the end, and thank you for staying in and listening to the talk.
So thank you very much, Jan. Very nice talk. So the ones that want to do some questions, please do the questions as soon as possible using the chat of the YouTube, and then I will read the questions to Jan. But waiting for the questions, I have one. You just briefly talk about the stability of the organic circuits. So can you comment a little bit more about this? What are the perspectives to have some stable circuits, or which are the conditions to?
Yeah. So we have to. I mean, there is, as I said, a lot of noise in it. This is also what we have learned when we work with electronic circuits with electric signals. And there are many of the things that we have applied there to get it to stabilize it and making sure that it stay within boundaries and so on, that can be applied into these circuits as well. The other thing I could mention is also and I didn't say anything about this, but within a single cell, it's quite limited still what you can do in terms of amount of logic. I think we are somewhere between 20, 30 gates or something like this. That's the limit. Because the cell has only so much capacity to actually stay alive and healthy by doing these things. And this is one of the really big questions, and we see it most pronounced in the simple systems from a computing point of view, those who are just doing fabrication processes of certain molecules. There you want them to stay alive as long as possible to build up the stuff. Another thing that I didn't address is that these cells are multiplying. So and every time they do, there is a chance of mutations, and you have to somehow know what that means. Typically, mutation will just mean that the function doesn't work, but how do you guarantee that the function doesn't turn harmful, especially if you want to use it as living pills or something like this for therapeutics? So there are a lot of practical issues that needs to be addressed.
Okay, thank you. So we have a question by Hushiur Nickel from Blue Spec, Massachusetts, USA. So what do you anticipate as the first practical problem that will be solved with these techniques?
So so I showed you the example with the psoriasis. Now today there are you can actually buy some engineered bacteria that is helping mitigating problems in your microbiome. So there are things. I mean, there's a lot of trials going on different things of actually trying to address issues of cancer, not just detecting, but also trying to let's say produce something that can fight against the cancer. So there are already practical problems, especially in the healthcare.
Thank you. So now a question by Jorge Science Noval from Institute of Microelectronics in Seville, Spain. So the method is very good for machine learning computing, semi and storage operations. Have you tried it?
So no. So I know of at least one paper trying to get using this to get a collection of cells being able to play tic-tac-toe. Just as a fun example, where you could trigger so you program the circuit in a way that you can change the functionality of them depending on some inputs. So yes, in principle, you could do this. I don't think that would make sense. And that's also why I put this disclaimer in the beginning. The switching times, and I should have mentioned that, so it's good you asked this question, is not on nano or millisecond scale. We're talking about seconds or minutes or more. So it's a very slow switching circuits. So I don't think you would rather tap into then neural network. I mean, real neurons, if you wanted to do something like this. On the other hand, machine learning has been used as a way to try to identify how to build the circuits. What is a good circuit to map to a certain function? We haven't looked into to that yet. We have tried to stay with this logic design philosophy that we know from VLSI design and other things.
Thank you. So now a question by Renan Marx from Federal University of South Mato Grosso this is in the center west of Brazil. Very nice talk. The simulation prototyping frameworks presented could also be used in researching of DNA strand displacement networks.
Yeah, so the microfluidic part of this, we actually have been considering looking into DNA strand displacement as a way to form logic functions. And there you can have aptamers to capture input molecules and triggering the onset of these strand displacement mappings. Now, for building the logic, it's very different from this one. So the whole design process would be very, very much different. But there are tools that could help you doing that part. But the back end of it would work. Yes.
Thank you. So another question by Hishur Nikhil. 3D machines and so on are universal. They can simulate themselves, and we can have layers of interpretation. Is this true in the biocomputing as well, or is it only first order?
Yeah. So that is that's a very good question. I think the way that it works has been shown to be Turing complete. So actually working as a universal machine. I haven't been thinking about trying to use it to simulate itself, and I think that's at the moment we will probably not be able to produce circuits big enough to actually do something like this. But it's a nice and fascinating idea. There has been, and there are some. I've seen some papers where they try to build sort of like a mechanical part of the way that the biology works with putting the things together and showing that this is actually a universal Turing machine.
Okay, so thank you very much. I'm seeing no more questions here in the chat.